Okay, so good evening, everybody. So we'll start the class because today we have a lot to cover. Um, just a minute. I think students would be joining now. Wait time. Okay, so this class. So I think we have 6 chapters. So because we have a lot of syllabus to cover for this week, so we'll be going a little quick and we might not go into each and every detail because we don't have that much of time. But if you have any problem, if you have read or if you are going to read see the online videos, recorded videos, you can ask me. So let's start. So this week it is just a minute. Yeah. Okay. Fine. So, in the last week, we have studied about the different kind of RNA processing events that happens after your transcription has taken place. So, when transcription takes place, your pre-mRNA is formed, and this pre-mRNA has to undergo several kinds of. Just a minute. Yeah. So after transcription, what is formed is a primary transcript. Okay, and this primary transcript undergoes several kinds of modifications, and those modification converts the primary transcript into a mature transcript. And we have studied about what kind of modifications. We have studied about splicing, we have studied about poly-A-tailing, and we have studied about capping. And apart from this, we have also studied about some kind of methylation events that happens that makes the RNA resistant to several nucleases enzymes. So this is what we have studied in our last class. Now in this week, it is about some alternative RNA processing and editing events that takes place. So apart from the one that are already known that we might have studied earlier as well, such as poly-A-tailing, splicing, capping, methylation, there are some other RNA processing events also that takes place. And what are those and how it helps the mRNA and why they are needed actually, that is all we are going to see in today's lecture. Okay, so first lecture is about the alternative splicing. So I don't know if you remember or not, but in the last class also we have discussed a bit about alternative splicing and why it is needed and how it actually takes place. Just a minute. Yeah, fine. So alternative splicing means conversion of pre-mRNA species into more than one type of mature RNA. Okay, from the same pre-mRNA, we are generating more than one type of mRNA, that is known as alternative splicing or alternative processing. Okay, in general it is known as alternative processing. So how this alternating processing can take place? Alternative, so for the students who have just joined, just a brief recap of what we have started. We have just started our lecture. So in this week we have to see the alternative RNA processing and editing events that takes place in the RNA molecule. So in that context we have just started with our lecture 1, which is the alternative splicing that takes place. So first we see that what is alternative processing. It is the conversion of pre-mRNA species into more than one type of mature mRNA. So what does I mean from this is that if this is your pre-mRNA, okay, this is your pre-mRNA, so it can give rise to more than one kind of mature mRNA. And not just mRNA, we can this alternative processing also holds true for tRNA also, we'll see later. So from the same pre-mRNA you are getting more than one type of mature mRNA. So this happens because of alternative processing, means there are more than one ways of processing this mRNA, and this alternative processing can be achieved by either alternative splicing or alternative poly-A-tailing. Okay, now first we'll see about alternative poly-A-tailing. That means, what does it means? So I'll just explain you in a different slide. Okay, this is your mRNA. This is your polyadenylation signal sequence. So if you remember, in your poly A tailing, what happens? There is a signal sequence, and suppose this is your signal sequence, okay, and some bases after 50 to 20 bases after your poly A polymerase, your enzyme comes, it cuts here, and then it attaches your poly A. This is what we have studied in our last class, that there is a signal sequence which is recognized by the enzyme. Then enzyme moves 50 to 20 base downstream of this signal sequence, and then it makes a cut, and then it ligates and attaches the poly A tail. This is what poly A tailing means. That means where this enzyme is going to attach the poly A tail depends on the signal sequence. Correct or not? Wherever the enzyme will find a signal sequence, it will recognize it, it will move 50 to 20 bases forward, and then it will make a cut, and then it will attach the poly A tail. So don't you think there can be a possibility that there are more than one signal sequence in the mRNA? Yes, there can be. So if in this mRNA you have signal sequence 1, and somewhere here you also have signal sequence 2, and let's say suppose here you also have signal sequence 3. So now if it recognizes this signal sequence 1, it will attach poly A somewhere here. If it recognizes signal sequence 2, attach poly A somewhere here. Signal sequence 3, attach poly A somewhere here. And we know that poly A tail is attached at the 3' end. That means the length of the mRNA would be different in all the three cases. So from the same primary transcript, depending on which signal sequence is recognized by the enzyme, your mature RNA would be different. Is it clear to you or not? Tell me. What I am trying to trying to tell, is it clear to everybody? Just write Y or N, yes or no, tell me. How different mature RNA can be produced from the same primary transcript, because there are more than one signal sequence present. So okay, so it's good that people are understanding. Okay, fine. So this is alternative splicing में first thing, that is alternative poly A tailing. Okay, this is alternative poly A attachment at different locations. Okay, I'm sorry for my bad handwriting. I'm not very used to using this writing pad, that's why. Anyways, now there are so many signal sequences. There is signal sequence 1, 2, 3. Now next question comes, that which signal sequence will be recognized. Are there any factors that decides that which signal sequence would be recognized? No, there are no such factors. But just like for the expression of genes, we have one dominant gene and when recessive gene. So when dominant gene is present along with recessive gene, the character which is shown by the dominant gene is only expressed. Okay, so what happens that suppose if there are three signal sequences, then one would be a very strongest signal sequence. Okay, so suppose signal sequence 1 is the strongest, that means it is the dominant. So by default this signal sequence will only be considered, and poly tails would be attached here, because this is the strongest signal sequence amongst the three of them. But because of any random event, now that random event can be anything, what can happen, that just a minute, yeah, But because of some any random event, what can happen, that signal sequence 1 is not recognized, and enzyme may recognize signal sequence 2 or signal sequence 3. So by default the strongest signal sequence is only recognized, but any random event can lead to the recognition of some other signal sequence, or other possibility is that these signal sequence are also recognized in a cell type specific manner. What does this mean? That suppose a particular mRNA, okay, that particular mRNA, in suppose it is in liver and brain, okay. Now in brain it has to be of a certain length 2, here it has to be of a greater length 1. So here signal sequence 1 might be recognized, here signal sequence 2 might be recognized. So it depends. So the recognition of signal sequence can be cell type specific also, or any random event might also lead to the generation of different recognition of different signal sequence. Okay, second thing is, so here what I have told you, that more than one poly A sites are present, and the stronger poly A site is used by default in the cell, but a factor may prevent stronger site being used in another cell. Okay, then we come to alternative splicing. This was about alternative poly A sites. Okay, now we'll see alternative splicing. What is alternative splicing? The generation of different mature mRNAs from a particular type of gene transcript can occur by varying the use of 5' and 3' slides in four ways. So alternative splicing can take place in four ways. First is different promoters, different poly A sites, retaining certain introns, and retaining or removing certain exons. So this different poly A site, it is similar to what we have just studied, that how your mRNA would be spliced also depends upon what poly A sites you have used. Suppose if you have used, suppose if there is exon number 1, exon number 2, exon number 3, okay, 3. Now if I have used this splice site, that means I have 1 and 2 exon sites. If I have used this splice site, sorry, polyadenylation site, then I have all 1, 2 and 3. If I have used just this one, this site, then I have only one. So depending on which poly A site you have used, your splicing will also be affected. Okay, then retaining certain introns and retaining or removing certain exons. Okay, so this I'll explain you. Renuka Devi, you have any question? Okay, fine. Okay, fine. Okay, fine, Renuka. So again, see, the first few slides where they will be talking about alternative splicing and alternative poly A tailing and some little bit about RNA editing. So that I will be explaining you, because this week you have a lot of things to cover. So I cannot tell you each and everything, but I will try to tell you the basic concept and logic which will help you to understand the theory when you will read it, or when you will reread the or re watch the videos, recorded videos. Okay, so there are certain mechanisms that sir has explained in the recorded videos that are very straightforward, or some, and very much examples, very more examples are also there. A lot of examples for RNA editing are there. So I cannot tell you each and every example in detail, because it will take more than 2 hours for me. I have to deal with questions also. So I'll tell you the basic logic behind it, and the examples are repetitive. So when you will watch the videos, you will again uh, recall what I have taught you in the class, then you will be able to understand the examples as well. Okay, now again, basic very important concept is alternative splicing. So this is your pre-mRNA. Now this is your exon 1, exon 2, exon 3. Fine. In between, you have okay, intron 1, intron 2 and intron 3. This is the basic now alternative splicing means splicing of what? Splicing of introns. So if this intron is spliced and this intron is spliced, only if I1 and I2 are spliced, then what will come together? This is not we are not considering right now. I1 and I2 are spliced, that means E1, E2, E3 both will combine. Okay, E1 will combine, E2 will combine, E3 will combine. Fine. If only I2 is spliced, only I2, then what will happen? E2 and E3 are combined. Fine. If only I1 is spliced, then E1 and E2 is combined. Okay. If I3 is spliced, then suppose if there is E4 or some other exon, then this E3 will combine with some other exon. So what I'm trying to tell is that depending on the combination in which the introns are combined, splicing out, your exons will be different. Your length of the mature mRNA would also be different. Here you can see the length would be greater. Here the length is smaller. Here also the length is smaller. Why? Because here 3 exons have combined. Here only 2 exons have combined. Here also 2 exons, and these are different exons have combined. So that means that from the same mRNAs, from the same pre-mRNA, you can have different combinations of mature mRNA. This is your alternative splicing. That means your from the same mRNA you can produce different mature mRNA. That means different types of proteins can be produced. Is it clear or not what I'm trying to tell? Just a minute. Okay. So that means because in our body the number of mRNA transcripts are way lower than the number of proteins that are present. That means that same mRNA is able to produce more than one kind of proteins. So how from same mRNA we can have more than one kind of proteins? Because of alternative splicing. That is why alternative splicing becomes so important, because this alternative splicing actually is helping in the production of different kinds of proteins from the same mRNA. Suppose the expression of all the 3 genes is needed, let's say in lungs. In eyes, E1 is not required. So therefore the splicing has taken in such a way that only E2 and E3 are there. Then suppose say in liver, E3 is not important. So splicing has occurred in such a way that only E1 and E2 are there. So the splicing from the same mRNA, you will have different kinds of combination of the mature mRNA, which will ultimately produce different kinds of genes. This is your alternative splicing. Then intron advantage. So now we see that prokaryotes, they do not have introns. Okay. And also prokaryotes do not have repetitive sequences as they are present in humans. So now why you think that humans have intronic regions, although they are removed only? Because these intronic regions helps to synthesize different proteins from the same mRNA. Okay, the question may come that if in a gene introns are getting removed, then why there is requirement for these introns at the first place? When we are already removing these introns in the mature mRNA transcript, then why they are needed at first place? Why the evolution has not taken place that there were no introns and just exon? It would have saved a lot of energy. It would have saved a lot of ATP. So why you think that I1, I2 and I3 are needed although they are getting spliced out in the mature form? Can you tell me? I just explained. Can you tell me? Anybody? Just why introns are needed? Why you think that the addition of introns in eukaryotic system has helped in its evolution? Can anybody
to,
tell me how
to
it has helped
generate
in? Yes.
to generate different proteins.
Yes. So how, what will be the advantage of the generation of different proteins? I mean in evolutionary scale. Yes, Kavita, you are right. Jahanara, you are also right. Variety of proteins. So that is why humans or eukaryotes, especially if we talk about humans, in even in eukaryotic scale also. I mean, yeast is also a eukaryote, and Homo sapiens are also eukaryotes. But our genetic makeup is even more complex than that of yeast, right? So this complex genetic makeup, this presence of introns, the presence of repetitive sequence, it has helped in the evolution of organisms in a larger scale, because we have so many different kinds of proteins that can be synthesized from the same mRNA molecule, from the same primary transcript, and those protein codes for different uh, uh, proteins that are needed for our body for evolution or for anything, that has helped us to survive in the longer run. So that is one important thing, that despite if you, I don't know whether you have ever thought of it or not, that anything in our body which is waste is, on the evolutionary scale, is always removed. Like I have told you that we have a tailbone, but we don't have a tail anymore because we don't need tail. Now the present body of Homo sapien does not require the need of a tail. So tailbone is there, but there is no tail now present, because if there would have been a tail, it would just have taken a lot more of energy to sustain it. So anything which is not needed and still to just make it happen would be energetically unfavorable. That is why tail is removed on the evolutionary scale from our body. But these introns are not removed on the evolutionary scale, although they are getting wiped out in the mature mRNA form, because these introns are helping to make different varieties of proteins. And not just that they help in making different varieties of proteins, I tell you, these introns have even a lot more different kind of functions, even more important function than the exonic regions, because these introns have non-coding RNAs. Okay. Also, um, these introns helps in the expressivity of the protein, expressivity of the mRNA. So that is what I have written over here. See, so we have discussed the first point, that is alternative splicing, and second point is expressivity of a gene is 100 folds higher if the cloned fragment also contains intron. So suppose if you are doing molecular cloning in your lab, and if you want to clone a fragment, this is a gene X that you want to clone in a vector. So if you have this gene, if this gene also contain intronic regions, then the expressivity would be higher than only if this gene has exonic regions. Can anybody tell me why the expressivity of a gene is increased if the clone fragment also contain introns? Anybody who can think of a answer? Anything, just any random guess. Can you think of a reason? Try to think.
Maybe introns help in alternative splicing now.
Okay, it helps in alternative splicing, and Kavita has said regulation. See, it helps in alternative splicing. That is one of the reason that why the expressivity is getting increased. But I am talking about in vitro. That means in lab. So in lab, I know that this I want, suppose a protein A, and I know to produce this protein A I need gene X. So even if it is doing alternative splicing and producing some other proteins B, C and D, I don't need them. I just need this A product, this A protein. So how the presence of introns will help in the product, will help in increasing the expression of this A protein? How it will help? Any uh, thoughts on this? Any reason, possible reason that you could think of? Okay, so I'll tell you. So these intronic sequences, as I told you, that they are not only help in alternative splicing, but these sequences have certain conserved domains or certain sequences, or even not even conserved also, that helps in the expressivity. How it helps in the expressivity? Because they increase the expression of that gene. This intronic region, it can regulate the expression of exonic region. So what does it mean is that if a certain gene X, it has to express, okay, so we need the exon of X, we need X exon. But if there is some intronic region just before it, let's say X dash, so X will be expressed only when exon is present. But how much it will express, it will depend on the intronic sequence. Whether it will express 10 times or 20 times or 100 times, this is governed by the intronic region. It will express for sure, but the level of expression, how much it will express, that will depend on the intronic regions, because intronic regions have certain sequences that govern the expression of the exonic regions, and that is the reason why the expressivity of a gene will increase if intronic regions are also present. Okay, so this is about the first class. Moreover, we have studied all these things about the first class. Now I just have a small two questions. Even if you don't know the answers, it's fine, but try to attempt it. So the first question is, average size of an exon is 150 nucleotide, 300 nucleotide, 1000 nucleotide, or 100 nucleotide. So this has not been discussed, but if you have any random guess, you can tell me. There is no logic behind it, it is just a thing to memorize. So do you know what is the average size of intron? Okay, fine. So Kavita, Jyoti, Tejaswini, another Jyoti, Anuya, Jahanara, Hemlata. So everybody has answered it very correctly. So the correct answer is 150 nucleotides. Good. Next question is, when pre-mRNA is spliced more than one way, it results in truncated RNA, incomplete RNA, RNA isoforms, or RNA concatemer. What does it result in? Okay. So, Jyoti, both the Jyotis, Kinnera, Satya, Jahanara. So, everybody has answered C. Tejaswini, C. RNA isoforms, correct? So, I just want to ask, so were these questions, like you have one online class, ma'am, don't turn screen. Okay, okay, I will not turn. Actually, some students, because when I am opening this screen, then I cannot see what you are answering. So, I have to shift the screen to see who all are answering me. That's why. So, Acha, tell me that in your Mondays class, in your Mondays online class, so what you have studied, which week lectures? This week or the previous weeks? Previous week? Week 4. So that means this is week 5. That means this you are studying for the first time. These things you are studying for the first time, right? Whatever I am teaching you. Okay, then it's really good that you know the answers. I just wanted to know that were these questions already discussed or you are answering it for the first time. If you are answering it for the first time, then it's very good to see that you know the answers. Okay, so the question was that when pre-mRNA is spliced more than one way, it will result in isoforms. Obviously, incomplete RNA or truncated RNA, it can happen, but it is not always the case. RNA concatemer. So, do you know what is the meaning of concatemer?
No,
Concatemer.
madam.
Yes, somebody was trying to tell. So what is the meaning of concatemer? So it means that multiple, if I talk about RNA concatemer, that means multiple molecules of RNA molecules, they are linked in series. They are not divided. They are not separated. It's like a chain. Just like a chain, you have a chain. We all wear chain like this. We are different. It's like this. It is a concatemer. So different molecules are linked together. They are not separated. They are not like this. They are like this. It is a concatemer. Okay. Now we come to implications of introns. We come to lecture 2, alternative RNA processing, implications of intron. So, up till now we have studied about alternative splicing, that two or more polypeptides can be derived from a single gene. From a single gene we can have many different types of polypeptides, because a single gene, when it will form a transcript, that transcript can be processed in many ways. So you can have many different types of mature RNA molecules, and ultimately those mature RNA molecules will generate different kinds of polypeptides or proteins. Then it is achieved by altering the pattern of exons that are spliced together. That is also we have just now learned, allows organism to carry a fewer genes in its genome. This is important, that in genome you need just few genes, and from few genes you can have many different proteins. Then different proteins can arise from a single mRNA. I think all these points must be clear to you now. Now this photo, why I have told you. So just to tell you, I'll just, yeah, just to show you that why introns are so important, that it not, I mean, in certain species, these, the presence of introns decides the gender, the sex of the genes also, the sex of the organism also. For example, sex in Drosophila is largely determined by alternative splicing. See, if 1 and 3, if 1 and 3 are spliced together, then it will lead to a female. If 1, 2 and 3 all are spliced together, then it will lead a male. That means this is the sex gene, and depending upon that which introns are getting spliced, it will be decided either it will be a female or male. Okay, sorry, these are the, okay, these are the exons. So if this 1 and 3 gets combined, so 1 and 3 are combining, it means if this and this are getting spliced, it will leads a female. Here also you see 1, 2 and 3, that means this and this, if they all are getting spliced, then you will have a male. So what kind of the splicing pattern, what kind of splicing that is happening, it is deciding the gender of the Drosophila also. So these introns are so important in evolution. It helps in determining the sex of a organism, in sex in Drosophila also. So this is very important. Okay. Yeah. Now some other, so these are, this image that I have taken and this example, so this is there in your recorded lectures, and some screenshots that I have shared with you, they are also in the recorded lectures and they are important. Now, alternative splicing can occur at specific developmental stages or in different cell types. What does it mean? That this alternative splicing can be needed in different cell types, just like we have studied by taking the example of liver, lung and eyes, how different splicing may generate different length of intron, which is exons, which is needed in different cells, or at specific developmental stages, just like we saw this example, that in the embryonic stage this kind of splicing is needed to decide the sex of the Drosophila. Then for example, so then see, in this week's lecture there are many examples, so not every example can be explained to you because of time constraint, but if you know the concept, and then when you will reread the lectures or re-watch the lectures, then those examples will automatically become clear to you. Okay. Now, so the calcitonin gene, it yields an mRNA that can synthesize calcitonin or calcitonin related peptides. So both things can be synthesized from one same mRNA. That means what they are saying, that calcitonin gene, due to alternative splicing, can synthesize either calcitonin or calcitonin related peptide, that means CGRP. This calcitonin is expressed in, is required in at thyroid gland, and this is required in brain. So these are the two proteins which have so different functions. See, from the same gene you have two different proteins, and these two proteins have very different functions. Another example is alpha tropomyosin. So the alpha tropomyosin mRNA, it has at least 8 differentially alternatively spliced alpha tropomyosin mRNA. This is also okay. Then again, this beta thalassemia. So in this whole lecture, beta thalassemia is discussed many times. Now, many defects, just a minute. Calcitonin parathyroid gland, ma'am. That is fine. See, I am telling you, what I am trying to tell you over here is that from the same gene, you can have different proteins which have which can have completely different functions. The functions can be completely opposite, or the functions can be completely uh, dissimilar. That is the logic over here, okay? And this all content, actually, this is, I have not written this content, this is all which has been provided by your sir only. Now, alternative splicing, again there is another example which is beta thalassemia. So we all know that there is a because of the defect in the beta globin genes, so beta thalassemia occurs, and some of the defects are caused by mutation in the sequences of mRNA which are required for intron recognition, and therefore results in abnormal processing. So what they are saying, this is your primary transcript of beta thalassemia gene. Okay, this is your primary transcript of beta thalassemia gene. Now, because of certain mutations that happens, the processing is disrupted, and because of the processing is disrupted, your beta thalassemia is caused. This is again an example: abnormal processing of the beta globin primary transcript. Okay, so there are many examples like this. Then again there is another example, I'll just tell you. See, again, this is a Drosophila gene. You see exon number 4, exon number 6, exon number 9, exon number 17. So this is your genomic DNA, or you can say pre-mRNA. Why genomic DNA and pre-mRNA are same? Because pre-mRNA also RNA processing has not taken place. So both introns or exons are there. And then it is mRNA, and then it is protein. So because of the alternative splicing of the 4 exons, 1, 2, 3, 4, so alternative splicing, it can lead to 38,000, almost 38,000 variations. You can see that this alternative splicing can produce so many variations, and these variants are important for wiring of the nervous system and for immune responses. Again an example. So all these examples just to show or tell you that why alternative splicing is so much important. It is determining the sex of an organism. It is from one mRNA you can have 38,000 different sets of proteins. See, so it is how much energy is getting saved. If only one mRNA would have been, so if only one protein would have been synthesized from one mRNA, then you would have needed 38,000 genes. But here, just from just one gene, you are getting 38,000 proteins. You see how much energy is getting saved, and how much genomic complexity is also getting reduced because of this alternative splicing event. Then just a quick revision. I thought to include this slide because I think this is a good slide to just in a very brief way explain you the splicing and the trans-esterification reaction that occurs. So basically, splicing involves trans-esterification reactions. Always remember this. So we have already studied that there is a 5' splice site, and there is a 3' splice site, and in between you have a link OH. So what happens, that this branch point, this OH is known as a branch point. So this branch point will first attack at the 5' splice site, and then there is a cut, and then there is a formation of this lariat structure, and then this OH will attack here, and then this lariat structure is removed, and then these two exons are joined together. So splicing occurs with the help of trans-esterification reaction, and you have 5' splice site, you have 3' splice site, and you have a branch point. This branch point first attacks at the 5' splice site, makes a cut, and then this spliced region, the cutted region, then it again attacks here and it cuts. And this structure is known as lariat structure. Then again, GTAG rule. What is this GTAG rule? So here we're discussing the earlier diagram that I showed is just to make you understand in a little bit more clearly about the splicing that happens. Okay, now what is this GTAG rule? Let's see. GTAG rule describes the presence of these constant dinucleotides at first and last two position of introns. Okay, so you see the first at the 5 primes, this is your intron, this is the 5 prime site of the intron, and this is the 3 prime site of the intron. Okay, so you see at the 5 prime site always GT is present, and here always AG is present. This is your GT-AG rule. So GT is present at the 5 prime site and AG is present at the 3 prime site. Okay. Then splice sites are the sequences immediately surrounding the exon intron boundaries. Okay. Splicing junctions are recognized only in the correct pairwise combinations. That means your branch point can recognize 5 prime splice site only if there is GT. So this GT-AG rule is important. At the 5 prime site GT should be there, at the 3 prime site AG should be there, or GUAG rule. Now we have two questions. I have to minimize so that I can see your answers also. So this completes our two lectures. Okay, now we'll just quickly see these questions and then we'll move to lecture 3. Okay. So the question is, introns are never seen in, it's very easy actually, eukaryotes, fungi, prokaryotes, plants. What is the answer? Introns are never seen in eukaryotes, fungi, prokaryotes, and plants. I'll wait for 1 minute and then I'll switch the screen to see your answers. Okay. Okay, I'll wait. Fine. So let's see. Okay. How many of you have answered? Okay. So Jyoti, Renuka, Satya, Anuya, Hemlata, Kavita, Jahanara, Tejaswini, Bhavya, Bhagyashri, Kinnira, Blessy, Trishala, Pooja, Swarna, and Vanaja. Okay. So you all Swarna. So C is the correct answer. Vanaja, you have answered A. Eukaryotes. No, but introns are present in eukaryotes. No, all that we have been just discussing up till now. We were talking about the splicing and the post transcriptional modifications and alternative splicing. So all this is it happens in eukaryotes, right? So why you think it is prokaryotes? Can you tell me, Vanja?
I think option A is prokaryotes, ma'am. Misunderstandingly put
Okay,
eukaryotes.
okay, fine. Now, next question is, percentage of alternatively spliced genes in humans is 10%, 65%, 35%, or 75%. I will wait for 1 minute so that you can see the question, and then I will switch the screen. So the percentage of alternatively spliced genes in humans: 10%, 65%, 35%, or 75%. Okay, so now I am switching the screen. Okay, so fine. Let's see what you all have answered. Jyoti, both the Jyotis, Hemlata, Janara, Trishala, Satya, Kinnara, Vanaja, Bhavya, Swarna, Blessy. So you all have answered correctly. The answer is D, that is percentage of alternatively spliced genes in humans is 75%. Correct. Good. I am very happy that you uh, people are answering correctly and able to understand. Fine. Now we'll come to lecture 3, that is splicing and pathology. So we won't go with the titles as per se, because uh, the content might be, you might get confused with the content if you will just go with the title. You just have to understand whatever concept is given in each and every slide, or in each and every slide even in your recorded videos. You just have to understand the content. Now, so there are different kinds of introns. This has also been studied, I think, discussed in previous class, but not in this much of depth. So there are four major types of introns, and they can be distinguished on the basis of their mechanism of splicing or their activity. For example, group 1 introns. So group A1 introns are known as self-splicing introns. So group 1 and group 2, this is important, not just for your this exam, but in general. Group 1 and group 2 introns, they are known as self splicing introns. Why they are known as self splicing introns? Because they do not need ATP. Group 1 and group 2 introns are known as self splicing introns because they do not require ATP. And then the introns which are present in the pre mRNA, that means the normal we are talking about, they require spliceosome, so they need ATP also. And in introns which are present in pre tRNA also, they also need ATP. So these group 1 introns are present in fungal mitochondria, plastids, and pre RNA of tetrahymena, and then group 2 introns are present in fungal mitochondria, plastids. So this is something to just you have to just memorize it. You just have to remember that group 1 and group 2 are self splicing introns because they do not require ATP. Then again, I just put up this slide. So in lecture if you go to lecture 3, so lecture 3 does has a little bit of mechanistic portion, otherwise things are to memorize. So you can just go, you have to go, actually this time you have to go, because especially for lecture 5 and 6, for lecture 5 and 6 I have not included that much part, because it is all mechanisms and examples that you have to go and read. Similar examples are getting repeated. Anyways, so now we have to see the pathologies which are resulting from aberrant splicing. So see, splicing is so important. It is not only generating different kinds of mRNAs. Introns not only generates different kinds of mRNA, mature mRNA. It not only helps in the expressibility of a protein, but if this mechanism is dysregulated, then it can also lead to several kind of pathological conditions. It can lead to diseased conditions also. So there are two major categories in which we can group the pathologies that are resulting from the aberrant splicing. The first one is mutations that affects the proteins that are involved in splicing, and second is mutation affecting a specific mRNA and disturbing the normal splicing pattern. Okay, so basically these are the examples. So if the proteins that are involved in splicing, if those are getting affected, if those are getting mutated, We can have diseases such as spinal muscular atrophy, retinitis pigmentosa, myotonic dystrophy, and if there are certain mutations that affects the mRNA, then you can have um, I mean a disease such as beta thalassemia, cystic fibrosis, Fraser syndrome, and these all diseases. Dementia can happen, Parkinson's can happen. All these again to memorize. Nothing to explain much in this slide. Just a minute, someone wants to join. Fine. Now comes some part that needs a little bit of explanation. So splicing in pre-tRNA molecule. So far we have studied about how splicing takes place in mRNA molecule. Okay. So in the last class also, in the last week's class also, and last to last week's class also, we have had few PPTs or few lectures where we have shown how splicing takes place in pre-mRNA molecule. But splicing also occurs in pre-tRNA molecule. And how it take, just a minute I have, yeah. So how splicing takes place in pre-tRNA molecule, that we have to see. So you see here, this is your pre-tRNA molecule, and this portion, this particular portion is the intronic region. So first what happens, that there is an endonuclease. So endonuclease means ase means enzyme which is going to cut. Nuclei means it will cut nucleic acids, that means RNA or DNA. Endo means inside, that means endonucleases are the enzymes which cut in the interior portion of the nucleotides, interior portion of DNA or RNA. So when endonuclease acts upon this, so what happens? See, from here, this much portion is removed, and little bit this portion also. So you are left with like this, which is 2,3 cyclic phosphate. Then what happens? Then ATP, because nucleotides are there, so you need ATP energy. Ligase, you need ligase for the joining. Then what happens? This portion would be joined, and this would come like this. This portion will come and join, and this portion would be like this. So you will have a spliced tRNA molecule, and this is your excised intron. So what happens? The non-coding intron sequences are excised from RNA transcript in the nucleus prior to the transport to cytoplasm, and introns in tRNA precursors are removed by concerted action of splicing endonuclease and ligases. These are the splicing endonuclease, and these are the ligase. Whereas introns in some rRNA precursors are spliced out autocatalytically with no catalytic protein involved. That means here, in the case of pre-tRNA, it is not a autocatalytic function. You require the help of certain endonuclease and certain ligases in the case of tRNA molecule, in the case of tRNA. Whereas in the case of ribosomal RNA molecule, the splicing happens by itself. There is an autocatalytic function. So no catalytic endonuclease or ligases are needed in the case of rRNA molecule. Okay, is it clear to everybody? Just a minute. Mm. Okay, fine. Now, another very important topic is R loop hybridization, which we will discuss in the next class, in the next lecture in this class only. in the next lecture. So this R loop hybridization is the technique which has helped in the discovery of introns, and this discovery of introns, discovery of split genes, has led to the Nobel Prize to Dr. Richard Robert and Dr. Philip Sharp. Okay. uh, just a minute, actually I have some very important this thing. Yeah. So before this, we will solve two questions and then we'll move to the next lecture where we will study about R loop hybridization in details. This is an important technique and this is also an interesting technique. Okay. Now questions. So first question is, rate of nucleotide synthesis is highest in humans, Drosophila, E. coli or fungi. So in which of the organism rate of nucleotide synthesis is highest? Humans, Drosophila, E. coli or fungi? So I will give you 1 minute to think and then I'll switch the screen. Okay, now I'll switch the screen. So many students have answered. Let's see. So the correct answer is okay. Yeah. So Janara, Vanaja, Kavita, Kinnara, Satya, Jyoti, Blessy, Chandana, Bhavya, Tejaswini. So you all have answered it correctly. The correct answer is E-Core 1. Now E. coli. So now even if you don't know the answer, how you can think that E. coli should be the correct answer? Because E. coli is a very simple system. It's a prokaryotic simple system. Humans. So when you have, as the complexity increases, it's a general rule that as complexity increases, the rate of synthesis will also increase. E. coli is a very simple organism, simple system. So obviously the rate of nucleotide would be highest. So a random guess you can also do even if you don't know the answer. Then next question is, key reaction in RNA splicing is, this we have discussed, condensation, hydrogen bonding, transesterification or coupling. What is the key reaction that happens in RNA splicing? 1 minute I'll give you, then I'll switch the screen. Okay, now I'll switch the screen. So many students have answered. Very good. I'm very happy that students are answering now. So from where it has started, I don't know, but I'll just say the names. So Tejaswini, Hemlata, Kavita. Achha, from here it has started. Okay, Hemlata, Anuya, then Bhavya, Jyoti, both the Jyotis, Renuka Devi, Jahanara, Supraja, Tejaswini, Vanja, Varaja, Chandana, Blessy, Satya, Swarna. So everybody has answered it correctly. So the correct answer is trans-esterification reaction we have just discussed. I'm sorry if I am unable to pronounce your name correctly. So I'm saying sorry in advance if I'm pronouncing your name incorrectly. Okay. Now, now we'll come to lecture 4 where we'll see RNA editing in detail. So before moving to RNA editing, so the main important points of this week lecture are first the alternative splicing and alternative poly A tailing that happens, how it happens, and why the alternative splicing is so important, because it helps in the determination of sex of certain organisms such as Drosophila. It helps in increasing the expressivity, and if it is dysregulated it can lead to many pathological conditions. And then we'll see how this introns were discovered, and then other important topic for this week is RNA editing. So RNA alternative splicing, polytailing, and RNA editing, these both are very important. This RNA editing, I would say that you have to go through sir's lecture, only then you will be able to understand. You won't be able to understand through my lecture, through my this class, because I have not included each and every detail of it. Because if I start explaining you the mechanism of each and every example, then to explain one mechanism only it will take 30 minutes for me. So I cannot include that such extensive elaborate detail in the class. But again, if you have any doubts you can ask me in the next class, and you can read through it. And also you have another online class on Monday in which you will study about these things only. So there also you can ask the doubt, or you can ask me also, whatever is fine with you. So this R loop hybridization is very important. In fact, even I didn't know about this R loop hybridization in such a great depth. I just uh, learned about it while uh, preparing my slides for you. So we all know introns. Now we have studied extensively about introns. But a very basic question comes, that how these introns were discovered. How they were able to see that these intronic sequences are also present within the DNA sequence. So this was discovered with the help of a technique which is known as R loop hybridization. So what this hybridization will tell you, just see over here in this, if you can see, this is your DNA, okay, and this is your RNA. Now this is when your RNA, for a temporary basis, it binds to your DNA, it forms an R loop. You see over here, partial denaturation of DNA under conditions that allow the RNA to displace the homologous DNA strand. So this is the double stranded DNA molecule. The partial DNA means not the complete strand is getting denatured, only a specific portion of the DNA is getting denatured. Okay, so partial denaturation of the DNA occurs, and what happens, see, this is the denaturation, this area shows denaturation, and because this partial denaturation when occurs, this RNA immediately comes and it binds over here and it forms R loop. This is DNA RNA duplex. Here it is DNA. This is also DNA. This is also DNA. Here this is DNA and this is RNA. So there is a formation of DNA RNA duplex. This is known as R loop. Okay, so what is written over here? The RNA pairs with the complementary strand of DNA, forming a RNA DNA duplex and leaving a single stranded region of DNA which is called as R loop. This is your R loop. Fine. This single stranded region of DNA, it describes the R loop. So now why we are studying about this? So in general, what should have happened? That if you have pre-mRNA, okay, I have to, yeah. So what I'll do is, I'll copy this here so that I can explain you what I want. Yeah. Now this is your pre-mRNA. And this pre-mRNA is formed from DNA. This is the DNA. From this pre-mRNA has been formed. Okay. Now, this pre-mRNA has exon number 1, exon number 2, exon number 3. Let's say these are exons 1, 2 and 3. Now, when this pre-mRNA is converted to mature mRNA, you only have these regions 1, 2 and 3. Now tell me, the length of mature mRNA, it would be greater than that of DNA, less than that of DNA, or more than that of DNA? Tell me, the length of mature RNA would be dash with that of DNA. Filling the blanks. Lesser than DNA,
Shorter than, lesser than.
lesser than DNA. So that means this mature mRNA would be of shorter length than that of DNA. Now, what was observed? Then, when this mature RNA was put to hybridize with the DNA molecule, see over here, when this mature mRNA was led to hybridize with the DNA molecule, this is the mRNA. So it could not hybridize completely with the DNA. See, so this is the region which it is able to hybridize. This all it is able to hybridize. Again, it could not hybridize this much portion. Again, it is able to hybridize this much. It could not hybridize this much portion. That means there were certain loops which were there. This is loop 1, loop 2, loop 3, loop 4. That means these are certain regions which are present in DNA, but they are not present in mRNA. That means when the mRNA is getting processed from primary mRNA to mature mRNA, these regions are getting removed because pre-mRNA was able to hybridize completely with the DNA. But when this pre-mRNA was converted into mature mRNA, then this mRNA was not able to hybridize completely. There were some regions that were looped out. So this led to the discovery that certain regions of the pre-mRNA, they get removed while they are processed to mature mRNA. And these removed regions are nothing but introns. And this is how, because of this R loop hybridization, the base is R loop hybridization, which says that RNA is able to hybridize with DNA. Because RNA is able to hybridize with DNA, that is the only reason why we are able to see that pre-mRNA is of similar length, mature mRNA is of different length, because they are able to hybridize. So because this, by employing the use of the technique of R loop hybridization, scientists were able to discover that there are certain regions which are getting looped out when mature mRNA is hybridized to DNA. Whereas primary mRNA, it can completely hybridize with DNA. That means these looped out regions are the intronic regions. So is it clear to everybody? The discovery R loop hybridization? Yes or no?
Yes, ma'am.
Okay, fine. So this is an important concept, a very interesting concept also. This R loop hybridization is a very interesting concept, and this concept has led to led to the Nobel Prize for Dr. Richard Roberts and Dr. Philip Sharp. Okay, and I'll tell you a very important this thing. Very interesting thing is such a coincidence that just two 2 to 3 days back, I think last week only, Dr. Richard Robert, he came to our university, like he came to University of Hyderabad, and he delivered a lecture on splicing, and he delivered a lecture on how what was the way by which led to the discovery of this splicing phenomena and this split genes phenomena. And see, the coincidence is that today I am teaching you this the same phenomena that has been discovered by Dr. Richard Robert. And just 2 3 days back I was attending his talk in my university. Anyways,
Great,
great,
ma'am.
ma'am. Yeah, it was actually great to in person hear a Nobel's laureate. It really motivates you a lot, and it really just energize you that despite of all the difficulties that we face, despite of all the apprehensions that we have to pursue research and go for higher studies, even though we do not get that much of credit and financial support also, why we have to and pursue science, and it just gives you a spark to even pursue science in depth. Anyways, so now we'll move to the another topic. So we have studied about R loop hybridization, and now we'll move to the other topic, which is RNA editing. So now what we are going to see, till now what we have seen, that whatever changes were happening here, in what is the difference between alternative processing and RNA editing. So RNA editing means that you are changing, specifically you are changing the sequence of the primary transcript, whereas the specific change of the sequence of primary transcript does not happen in the case of processing. In processing, you are removing a particular segment. Okay, in RNA processing, what happens? Either you are adding some poly-A tail, or you are putting a methyl cap, or you are removing exonic or entonic regions, but you are not changing the sequence of the primary transcript. The primary transcript sequence, when it is changed, then it is known as RNA editing. That means you are specifically changing the primary transcript sequence. Okay, now this sequence can be changed in two ways. First way is base modification, and second one is base insertion or deletion. Now, base modification means deamination reactions. Okay, so adenosine and cytosine. So cytosine is converted into uracil. C to U change, this is known as deamination reaction. And the uridine, the insertion of uridine base or deletion, that is your insertion or deletion. So that means there are two mechanisms for gene editing. The first one is uridine insertion or deletion, this one, and second one is base modification, that means site specific deamination of adenosine and cytosine. Now, can anybody tell me, in general, if we talk about mutations, okay, what is more deleterious? What is more what which is more harmful for the cell? Insertion or deletion, or base modification? If we talk about mutations, or even in the context of RNA editing, which can lead to greater changes, let's say, insertion or deletion or base modifications? Can anybody tell me? Which has a higher impact, insertion deletions or base modifications? So Janara is saying deletion, Jyoti is saying both, base modification, Renuka is saying base modification, and Kavita is saying two deletion. Okay, now I'll tell you, I'll give you an example, and by this example, not only your RNA editing concept would be clear, your concept for mutations would also be clear. Okay, because what are these are nothing but mutations. I don't know if you have studied or not, but you have a specific topic of mutations in molecular biology, where you study the different types of mutations that happens: insertion, deletion mutation, frame shift mutations, and everything you study. This is the same concept. Now see, it's very interesting actually. Okay, so this is your sequence: AUG, GUC, CCA, TTG. Suppose this codes for methionine. Suppose I'm not saying this is GUG codes for arginine. Let's just imagine that it codes for arginine. Okay, this codes for any other X protein, and uh, This is TUG, okay, and this goes for any amino acid Y, let's say. Now what happens, that base modification occurs. So this G is converted to some other, any other base. Say it is converted to T. I'm not talking about deamination reaction specifically. I'm just taking that this base has been changed to T. So what will happen? This arginine would not be synthesized. All other rest would be synthesized. Okay. And now if we talk in context of base modification, suppose C is modified to U. So now it, earlier CCA used to code for X and UCA would code for some other protein Y. So this X protein is getting affected. Nothing else is getting affected. Okay. Now this is your base modification. This is your base modification. So this base modification will only affect that particular codon of which the base is getting affected, right? Now I say insertion or deletion. Suppose I say this U is deleted. So what will happen? AUG, 3 codons, it will code for methionine. Okay, this U is deleted, so there it was GUG. So what will happen? GGC, CA T and UC are left, UG are left. So now you see, because of the deletion of this U, the entire frame of the codon is changed. Has it changed or not? Because for the synthesis of one amino acid, you need 3 codons. So if this U is deleted, then you have GGC, then CAT, and then this UG are left. If something else is there, then suppose any other N nucleotide will come. So now you tell me which will have more deleterious effect or more pronounced effect, insertion or deletions or base modifications. Now can you tell me? Insertion, deletion?
Deletion.
Yes, yes. Jahanara, you were saying right.
Deletion.
Yes. So insertion or deletions will have more deleterious effect. Same thing goes with insertion also. Here we talked about deletion. Let's say 3 U's are added. Sorry, 2 U's are added, let's say. Here or somewhere here, if you add 2 U's. So AUG, earlier I'll just make it uh, better, so it is, and if suppose it is AUG, CCT or CCU, because we're talking about RNA, and UAG, suppose. Now here in this place 2 U's are added. So what will happen? AUG, then CUU, CUU and AG. Again you see entire frame has changed. So this means that insertion and deletion will always have a greater impact in comparison to base modifications, because base modification will only affect that particular codon of which base is getting affected, but insertion and deletion will affect all the codons that are downstream. All the codons, the entire frame would be changed. That is why it is no, in mutation terms it is known as frame shift mutation, because the entire frame gets shifted, entire frame gets changed. That is why it is also in mutation terms it is known as a frame shift mutation. Okay, so here we are talking about editing. So what I am saying, that editing can be achieved by two ways. Editing means that you are changing the sequence. Editing can be achieved by two ways. First is base ka modification, that means the deamination of cytosine or deamination of adenosine, or insertion or deletion of uridine. Now let's see. Yeah, important slide. This is important slide because it will clear your concept, and then if your concept is clear, then you can see n number of examples and you can learn about it. That is not a problem. This is important. I'll tell you about it. Yeah. See insertion and deletion, how it happens. So uses a guide RNA that is encoded elsewhere in the genome, and part of the guide RNA is complementary to the mRNA in the vicinity of editing. So you see here, um, this blue one is your guide RNA. Okay, so you see this much portion of guide RNA, it is complementary to the already existing RNA. Now here, just a minute. Yeah, see this portion and this portion, it is complementary, and here your insertion has to happen. So what the guide RNA do, the guide RNA first it will bind to the RNA sequence here, to the complementary sequence, then an endonuclease will come and endonuclease will cut here. Okay, here it will cut. At this point endonuclease will cut. Now what will happen, you are left with this. Then there is an enzyme which terminal uridine transferase it comes, which attaches uridine residues are attached over here. Okay, so now this cut portion, two U's are attached, and when two U's are attached, this guide RNA has AA which is complementary to U, and then it will keep on synthesizing. So you see how cleverly extra two user attached, extra two user inserted over here, because TUTS, terminal uridine transferase enzyme, it attaches to U here. Then comes deletion. So here there is attachment of 2 uridine, and deletion, there is deletion of 3 uridine residues. See over here, again this much portion is complementary, again there would be a cut, and these 3 U's are removed with the help of U exonuclease. Okay, these 3 U's are removed, you have only A left now. Now this guide RNA is made in such a way that it is complementary with the A, there is U. Okay, and this will again go on. So in this way you can see that either you can either you can add 2U or delete 3U's. This is your insertion and this is your deletion. Then comes your base modification. So deamination means what? Removal of the amino group. So amino group can be removed from bases which has amino group. Okay, so cytidine and adenosine, these two bases have amino group in it. So you see, cytidine, this is your amino group. When amino group is removed, it gets converted into uridine. So cytosine is converted into uracil, or cytidine is converted into uridine. And you see adenosine, this is the amino group. When this amino group is removed, it becomes inosine. And this happens with the help of the enzyme deaminase. So this is what the two kinds of modifications, yeah, these are the two kinds of modifications that happens. First is insertion or deletion, and the second one is base modification. Is it clear to everybody?
Yes, ma'am.
Okay. If you have any question in this, you can ask me. If there is
Yes,
any
ma'am.
doubt. Yes? Tejaswini, you want to ask something?
No, ma'am.
Okay, fine. I hope it is clear to you. So this concept is very important to understand for RNA editing. Okay, so we have example again. So now, so if you will go through your online recorded lectures, you will see that there are a lot many recorded examples. A lot many examples are there. One such example that I have taken, editing of apolipoprotein B. Yeah, you see, this is the unedited mRNA. It has CAA. CAA. Nothing happens, no RNA editing. It is 4563 amino acid long apoprotein is there. But when deamination of cytosine occurs and it is converted into uracil, UAA. Now UAA, UAG, UGA, these are the stop codons. Now what has happened? That because of the deamination of cytosine, it is converted into uracil and now it becomes from CAA, now it became UAA. So that means now it became a stop codon. And because it became a stop codon, it will stop the synthesis of the protein. So now this protein would only be 2000 amino acid long. So it would be half of the length of the protein. Just because of one single, you see the power of this substitution um reaction, the power of this base modification reaction. Such a powerful mechanism that it can reduce the length of the protein by half. There it is CA. Just one change of C to U and it is now apolipoprotein which is of half of the size. So this is the example of RNA editing. At how RNA editing can help in the uh, formation of different length or even different type of proteins. Okay. Here there is coming a different length because it is getting modified to a stop codon. It is not always that a different length would be produced, but here it is stop codon, so different length. Okay, so now we'll quickly see the questions. These are just true or false questions. So the first question is, R loop hybridization pave way for the detection of split genes. So split genes are nothing but the spliced genes or the introns. Okay, so it is true or false? R loop hybridization paved way for the detection of split genes. True or false? Tell me. Kavita is saying true. Jahanara is saying true. Any other responses? Okay. Vanja, Swarna, Kinnara, Tejaswini, Satya. So you all are true. Yes, the answer is true. So R loop hybridization helps in the detection of split genes. Now, any base in an RNA can undergo editing. Any base can undergo editing. Is it true or false? Any base in an RNA can undergo editing. True or false? Manja and Janara is saying true. Okay. Kavita is saying false. Okay. I want more participation. Jyoti is saying true. Fine. The answer is false. Why? Because we are talking about any base in an RNA can undergo editing. That means we are talking about base modifications. Okay. So in base modification, only adenosine and cytosine can undergo editing because deamination is the kind of modification that occurs. I just told you now that deamination is the removal of the amino group, and amino group can be removed from the base that has the amino group, and that is adenosine and cytosine. So adenosine is converted into inosine and cytosine is converted into uracil, or cytidine is converted into uridine. So not any base can undergo editing, only A and C can undergo editing. And even if we talk about insertion or deletion, the insertion and deletion of only uridine takes place. Only uridine is either inserted or deleted in the case of RNA editing. We are not talking about normal mutations. In mutations, any base can be inserted or any base can be deleted or modified. Here we are specifically talking about RNA editing. In RNA editing, addition and deletion of only uridine takes place and modification of either A or C takes place. So it's not any base that can undergo editing, it only A, C and U can undergo editing, and that is also different kind of editings. Is it clear to everybody, this question? Jyoti, is it clear to you? Okay. Now next question is, mRNA alternate splicing always brings truncated protein. True or false? Think. I'll give you a minute to think. mRNA alternate splicing always brings truncated protein. True or false? Okay, I am switching the screen. So a lot of answers are already there. Let me see. Okay. Fine, Jyoti. Then okay, Vanja, Kavita, Swarna, Kinnara, uh, Anuya, Jyoti. So you all have answered it very correctly. It is false. False. Only if the alternate splicing brings a truncated, stop codon, then only it can be a truncated protein. Not always that mRNA alternate splicing it brings a truncated protein. So this is false. Fine. Now we'll come to lecture 5. So in lecture 5 and lecture 6, there are basically all the mechanisms, variety of mechanisms which is beyond the scope of this 2 hours class to make you understand. So again I am telling you that I have taught you enough of the basics that are needed to learn the in depth knowledge of this particular week's lecture. So and I'm sure that this much of, if you could able to study or learn or understand the concepts that I was trying to teach today in class, then it would be easier for you to understand the mechanisms or different examples that are given in the copy. Okay, so we'll just see the basic crutch of what you have to study when you just revisit the lectures. So in lecture 5 they have talked about the relevance of RNA editing. So what are the functions of RNA editing? So there are certain examples that they have given. So they have given AI editing, that means adenosine to inosine editing, that takes place in the glutamate receptor GLUR2. And then there is another example that they have given, ADAR ADAR2 editing in the central nervous system, and then editing of apolipoprotein BmRNA RNA. So these are the some examples that you have to, these are the examples which shows the relevance of RNA editing. These are just the examples, you have to go and you have to just see them. Then always remember that RNA editing enzymes that acts on double stranded RNA and not on single stranded RNA. This is important to learn, that RNA editing enzymes acts on double stranded RNA. Only then they will be able to make a change, obviously. If they are single stranded, if there is no complementary activity that is happening, then there is no use. Then in lecture C again you have many different relevance in immunology. So in lecture 5 you have these examples that I have just quoted, and some basic mechanism of how this AI editing takes place, how other editing takes place. It is not that much important, but still you have to have an idea. Must have an idea. The basics you can just read, and you can just let yourself know. Then in lecture 6 we have the uh, uh, relevance of RNA editing in immunology. So what is the relevance? Just I have told you that one single mRNA, one single mRNA can lead to the formation of different proteins. Okay, and this is for mRNA. If we talk about tRNA, because just like RNA alternative splicing occurs in the case of mRNA and tRNA both, in the similar way RNA editing also occurs in mRNA and tRNA both. So how this tRNA editing has relevance in immunology? Because we have just 30 tRNA molecules and we have 61 codons. So how this would be achieved? Because of RNA editing, because of tRNA editing. Now you see this in this figure, that if you can see, this is such an important figure. You see, you have, these are the four bases: A, TGC or AUGC, UG, UCAG. Their combination can make different kinds of codons, which can make different kinds of amino acids. See, UUU, UUC, UUU, UUC, UUA. How you have to read it? See, UCAG is written over here also. So UUU, then UUC, so UUC, then UUA, then UUG. Like this you have to read it. Now how to read this? UCU, UCC, UCA, UCG. Like this you have to read. This will tell the first letter, this will tell the second letter, and this will tell the third letter. Okay, so like this you should why I am telling you, this table is very important, guys, for your competitive examinations also, and for in any matter in general. This table is very important for you to learn. So when we talk about the relevance of RNA editing in immunology, so we know that here we are talking about specifically tRNA editing. So always remember that whatever kind of post transcriptional processing, whether it is RNA editing or it is alternative processing, these both are helping in generating diversity. The overall word is diversity. This all is helping in generation of diversity. How much diverse different kinds of proteins it can be produced, how much different kinds of mRNAs it can be synthesized from one single mRNA, either by splicing or polyetailing or capping, or by modifying a single base. A single base is modified, that can also lead to generation of different diversity. And those examples you can see here, that how AI editing in glutamate receptor it helps, how ADAR in CNS central nervous system it helps. You see over here, see, this is exon 3 and 4, and here both are A and A. Okay, just a minute. See, this is exon number 3 and 4, and this is exon number 5. In between you have A and A. Both A's are there. So this is your unedited, full length, functional. Now if deamination of adenosine to inosine takes place, so A is converted to I. Here you see, A is converted to I. Here editing has taken place. Now this edited becomes non-functional. So just because of one editing, this truncated mRNA, it has become non-functional. Okay, so this is your non-functional Adar. So just to tell you that the modification of just one base, how much difference it can make. Is it clear? This was the example for Adar in central nervous system. Similarly, there are many other examples of editing of apolipoprotein on B, apolipoprotein B on mRNA, and AI editing in glutamate receptor. So these are just the examples that you can just go through and have a gist of it. Important thing is here to have a gist of all the examples. Okay. Now we'll see the questions again. Prokaryotic RNA polymerase has only one subunit. True or false? Prokaryotic RNA polymerase has only one subunit. True or false? This question we have dealt earlier also. So Renuka has answered false. Tejaswini, Anuya, Satya, Kavita. So many have answered Jyoti, Yuvranjani. So many have answered it false. This is the correct answer because we have already studied that it has alpha 2 subunits, beta, beta dash, and omega. So there are not one subunit. There are so many. Sorry, sigma. There are so many subunits. Then mRNA alternate splicing always brings truncated proteins. True or false? Does alternative splicing always brings us truncated protein? True or false? So Jahanara, Yuvranjani, Tejaswini, Renuka Devi, and I'm sorry I'm unable to pronounce your name. I'll say you NG. So yes, so you all have answered it correctly that mRNA alternate splicing does not always bring out truncated proteins. Then next question. Which of the following genes show editing in their mRNA? Apolipoprotein B, glutamate receptor, cytochrome oxidase 2 gene, all of the above. So you might not know the answer of this question uh, as of now, but just try to answer it. Which of the following genes show editing in their mRNA molecule? 1 minute for you to think. Okay, now I'm switching the screen. Okay, so many have answered. Jyoti has answered D, Vanaja has answered A, Kalyani has answered A, Jayanara has answered A, Anuya B. Okay, so the correct answer is D, all of the above. That means all of the following genes, whether it is cytochrome oxidase, glutamate receptor, apolipo, we have already seen one example. So these all show the editing. The next question is all the following statements are correct about the RNA editing except. So you have to identify the incorrect statement. Information changes at the level of mRNA. In mammals mitochondria, RNA editing is a common pathway to control gene expression. RNA editing occurs through base substitution in the mRNA. RNA editing occurs through systematic insertion and deletion. Try to answer, try to pick the point which is least connected with the question. I'll give you 1 minute to think, which is least related. Think, and then I'll switch the screen. I'm not switching the screen so that you can see the question. Okay, now I'm switching the screen, okay? Fine. So, Bhavya, D, Tejaswini, Satya, Renuka, Anuya, uh, Jahanara, Vanaja, Satya, Jyoti. So, they all have answered D. So, okay, I understand your concern that why you have chosen D. So, first point is information change occur at the level of mRNA, correct. RNA editing occurs through base substitution, yes. RNA editing occurs through systematic insertion and deletion. This is actually correct, and the incorrect statement is B. Now what can be your question? That here it is written deletion basis, and just now we have seen that in insertion and deletion of only uridine takes place. Then how this can be the correct statement and this can be the incorrect statement? See, now when you deal with multiple choice questions, that is what you have to see. You have to select the statement which is most appropriate with in context of the question. If there would have been another option, E, here it would have been written insertion and deletion of uridine, then this would have become the correct statement and this would have become the incorrect statement. But here in these options, this is more correct in comparison to this. This is comparative. Comparatively, this is more correct. This is absolutely wrong. In mammals mitochondria, this is not true that in mammals mitochondria RNA editing is a common pathway. This is an incorrect statement. Why? Because mitochondria, mammals genome kind of reflects a prokaryotic system. If you have studied that some of the prokaryotic rims have transferred into the eukaryotic mitochondria and chloroplast. So mitochondria and chloroplast genome kind of reflects like a prokaryotic genome. And if it reflects like a prokaryotic genome, then there is no point of having any kind of post-transcriptional modifications, whether it is RNA editing or any other kind of modifications. So this statement becomes completely incorrect. So in comparison to this statement is more correct. This is like comparative. So when you have questions like MCQ, you have to see the one which is the most correct in context of the options that are given to you. Is it clear to you or not what I'm trying to tell over here?
Yes, ma'am.
Okay, fine. Just a minute. Now we'll come to the next question. The RNA editing through base substitution, okay, is a result of deamination of adenosine to become inosine, deamination of cytidine to become uridine, deamination of guanosine to become inosine, or D, both A and B. What is the correct option? I'll give you 1 minute to think about it. Think. 1 minute to think, and then I'll switch the screen. Think about what should be the answer. Then I'll switch. Okay, now I'll switch the screen. Sorry. Yeah. Okay. So Janvi, Trishala, Kalyani, Bhavya, Anuya, Kavita, uh, Jahanara, Renuka Devi, Tejaswini. Okay, so there are many people who have answered, and you have answered it correctly, both A and B. So basically, when we talk about base substitution, we have talked that base substitution of adenosine and cytidine takes place, because we are talking about what kind of base substitution it occurs. There is deamination, and deamination, as I already told you, is the removal of the amino group, and amino group can be removed from the bases that has an amino group. So adenine has an amino group, and cytosine or cytidine also has an amino group. So when the amino group from cytidine is removed, it is converted into uridine, and when the amino group of adenosine is removed, it converted into inosine. Just to recapitulate, this was the figure. You can see this is cytidine, it is converted into uridine. This is the amino group, this amino group is getting removed. Now there is double bond O over here. Similarly, adenosine has an amino group, this amino group is getting removed, and now it is converted into inosine. This is deamination. That means D means removal, amination means removal of the amino group. This is the base modification that has occurred. Okay. Next question is, during RNA editing in apolipoprotein B gene, deamination of which takes place? You might not know because you haven't studied the example, but just try to answer, then anyways I'm going to tell you the correct answer. Just try to answer the question. Someone has popped up. Okay, sorry. So you're giving the answers A. So Jyoti has answered as A. Okay, fine. Let's see what others have to answer. I'll wait for a minute and then I'll switch the screen. Meanwhile, you can think of the answer. Okay, I'm going to switch. Last few seconds to think. Okay, fine. Let's see. So Trishala, Anuya, Vanaja, Janara, Jyoti, they all have answered A, and it's very good that you know. So A is the correct answer. This means that you have studied the lectures. It's good. I'm very happy. So during RNA editing in apolipoprotein B, deamination of cytosine takes place and it is converted into uridine. Okay. Now the last question for today's class: which best describes alternative splicing? Using multiple proteins to create one gene, using one gene to make multiple proteins, using multiple proteins to multiple genes, or using one mRNA to make multiple proteins. Think about it. I'll give you 2 minutes for this question. Last question, 2 minutes. Think. What can be the answer? Think. 1 more minute to think for the question. See, the options can be confusing. You might think that you know the answer, but you have to be very careful. You have to look very carefully, especially for the two options. I won't tell you which two options.
Option B and D are looking alike, ma'am.
Yes, that is what I was telling, that option B and option D would look very much alike to each other. So that is where the confusion will come. See, let us discuss first. I'll see that whether somebody has answered or not. So yeah, so people have mostly taken B or D. Fine, I understand. So using multiple proteins to create one gene, it is absolutely wrong. We know this. Using multiple proteins to create multiple genes, again it is very wrong. Now here comes using one gene to make multiple proteins, or using one mRNA to make multiple proteins. Now, whether it should be gene or mRNA, that is the question over here, right? So, when we talk about alternate splicing, so we are talking about what? We are talking about a phenomena that occurs at a DNA level or RNA level. Tell me, this phenomena occurs at DNA level or RNA level?
RNA, ma'am.
RNA level.
RNA level. So when this phenomena occurs at RNA level, then can we use the term gene over here? No, because gene term would be used when we describe anything at the DNA level. So when we are saying that RNA level, the phenomena occurs at RNA level, so that means we are talking about that one mRNA which is used to make multiple proteins, because this would be more specific. Although that gene has only made mRNA, but you cannot just you have to be more specific. You have to see which makes the most sense here, because we're talking about a phenomena which occurs at RNA level. That is why we have to use, or we have to select option D, using one mRNA, because gene option would be used if we talk about anything that occurs at the DNA level. Is it clear to you? Is it clear to everybody?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Fine. So with this we'll stop today's class. And so in this today's class we have studied about some things which are different and unique. Alternative RNA processing and editing. So we talk about, when we talk about post transcriptional modifications, we say that okay, post transcriptional modifications means splicing, poly A tailing, capping. But these phenomena are different, that we might not have studied earlier or not in such great depth. But now, with the help of today's lecture and how introns are formed, how why introns are so important, how some same mRNA can lead to the formation of different proteins, and how even the substitution or deletion or addition of one base can lead to so much vast change in the cell. That is all we have studied. So I hope you have liked today's lecture, and if you have any query, can you display second last question, ma'am? Okay, Kavita has asked me to display second last question. I'll display. Yeah, this is the second last question, you can see. So I hope that you were able to learn at least something from today's class. And definitely, if you again go and re-watch the videos, or even if you're watching it for the first time, you'll watch the videos, you will be able to understand the context in what we were talking about, and at least a gist of the whole lecture. So with this we'll end today's lecture, and now we'll see next week. Okay, so there is one question. Ma'am, is there any specific condition undergoes alternate splicing? So yes, there are specific condition, Anuya. So specific conditions are, suppose these alternate splicing, so what introns are getting spliced depends upon what exons has to be joined. And what exons has to be joined, it depends upon what exons has to be expressed. And what exons has to be expressed depends on which cell we are talking about. So this alternate splicing is cell type specific, that in eyes and in liver same gene are there, same gene is there in eyes also, same gene is there in liver also, but what exons will be expressed in eyes would be different from that of liver, and that would be governed with one of the phenomena that governs is alternate splicing, not the only phenomena, but one of the phenomena that governs. Also, some aberrant changes, some aberrant mutational changes can also lead to alternate splicing, which can lead to diseased condition. That is a different thing. Okay, so I hope I was able to answer your question. So now we'll end today's class, and if you have any doubts, you can always ask me, or you can ask me in the next, the next class also. So okay then, bye bye all, have a happy week.